All right. Good evening, everybody. Welcome to Taxonomy Tuesday, where we look at each other's finds from the past weeks and days and try to help each other identify them. Show and tell style, or we could go. You could send them into Taxonomy Tuesday, and I could show them from my computer. Just put your name in the chat if you want to show them from your computer. And who wants to go first? I go to the email. Dave sent some. Hunter, Douglas, Brandon. Nobody?
Let's just start with the first person that sent an email.
Well, that be Dave.
I can go if he does not want to go yet.
Did he leave?
I'd be happy to go. I'm heading to New York, New Jersey in the morning, so to do mine first would be good because I'm kind of running around.
Oh, I'm sorry. Were you asking me to go? If Hunter wants to go, that's fine. Because I see my thing here.
Either way.
There we go. For Hunter's traveling.
Yeah, if you're pressed for time, Hunter, you go.
Yeah. Okay, thank you. I'm gonna
Sure.
stay, but I'll be like, running around the house at the same time. So
Can you see it?
yes. Yeah. So this is a really small Ramaria type mushroom that I've been seeing a lot. It looks very Ramaria-ish, like when fresh, but it kind of quickly turns to brown and more kind of stringy. So, but it's obviously related, and I don't know much more. I do have some spores, and I was able to get a spore print very easily, which was good. But I haven't had any time to really look deeper into it, nor even look and see if anyone, I don't think anyone put a name on this. It looks like no. So are you able to, oh, there we go. Okay. And if we can blow those up.
Well,
So I would describe these as, are these fusiform?
Maybe, but the queue is rather large. It's looks like a queue of well over two or three, somewhere in there. That's a Ramaria characteristic. Well, not for all the Ramaria species. I think the thing that you're really sort of dancing around here is whether you have a Ramaria or a Clavulina, which is a split off genus from Ramaria, I believe. And you're showing here some sort of conifer cone. Is that a spruce cone, Norway spruce or something?
White pine?
Oh, it's a white pine. Duh. I see hundreds of those and I didn't recognize it. Yeah. So the Clavulina tend to grow in needle duff. I'm not sure if they're saprobic or mycorrhizal, but
Okay.
I think they have shorter spores than Ramaria. I forget. I think either way, you're going to get like a yellow spore print.
Mm-hmm. I really didn't see much of a print, but I could see it under the microscope. You know what I mean?
Yeah.
I just did it on a slide, laid it on a slide, and it it was light colored, but not real dense to even actually be able to tell if it was white or cream or yellow or
Oh, okay.
yeah. But
And
I have it.
I think the Clavulina also have branches that terminate in more of a tapered sort of way. These look kind of blunt.
Yeah, and they're really like tightly packed too. You know,
Yeah, this looks like a Ramaria to me, but it's those Clavulina are pretty tricky. They're colored like this, maybe a little darker, maybe a little more ochre than sort of a little more lively yellow color. I think this is a Ramaria. That would be my guess.
okay, mine too, but yeah. Huh. All right.
And after that, even if you know it's a Ramaria, one time at a nymph, there was a Ramaria on the table, and Noah Siegel was there, and Noah is like one of the best mushroom identifiers in the world, really.
Right.
And I picked it up and showed it to him, and he laughed and said, "Good luck." So
Um,
was this growing on the ground or on wood? It was on the ground, right?
it was brown, yeah. Then there were a few others, and it's pretty small. The others were slightly bigger, but yeah, in that pine litter.
Yeah, I agree. It's probably a Ramaria. You know, if I was on MO, I would probably call just Ramaria promising. But yeah, getting to a species, I don't even know how to do that really.
Yeah, there was that one guy studying those. So I did keep it, and I should probably share it on iNat too, which I didn't. I'm honestly only putting these up for us, and I should care more about other reasons to put them up on these venues. But I right now it's like I just like to try to get them up here for ease of showing them for Tuesday night. So, but
Yeah,
yeah,
you should. You find cool stuff. Upload it onto, you know, add more data points.
yeah, I need to maybe yeah, maybe. Sometimes I don't know. I just yeah, I'm funny about the location stuff. I just not that comfortable with. Like if you look at my iNat thing, it looks like I'm a drug dealer making runs from Wisconsin down to the Gulf Coast, like and back and forth and back and forth and back and forth. And so it's just I don't know. I just feel kind of funny about that. But
Yeah, that's fair.
yeah,
Clearly, everyone on iNet knows exactly where I live
right.
Yeah,
from all the ones I find in my yard.
Yeah, so funny. All right. Well, anyways, and then really most of it is just time constraints, just doing the best I can. But yeah. So anyways, we can move on. I got a couple others. At least I did some microscopy this week, which made me happy. So another coral mushroom, and I'm, just put it in Clavulina. I am not sure what to call it. And I just didn't even give it any thought, but I did do the spores on it too, I believe. And it kind of reminded me of the Cerrena, but when I did take the time to look that one up, and it doesn't look like that. So there's what is it, cristata or
Yeah,
something?
that's what it is.
Oh, okay.
Yeah,
Okay.
Cineraria is actually a cristata that's infected by a secondary fungal growth.
Oh,
At least
okay.
I think that's the current hypothesis on the gray ones.
They had a great
It seemed
photo.
like the tips were more blunt on that as well, like more.
They probably erode when the fungus infects the fruit body.
Yeah,
Okay.
It probably changes the tips. So see how the tips are flattened and pointed?
Uh-huh.
Yeah, that's a cristata. Coralloides is another name that they can't seem to agree on, I guess. I
I was
suppose there's a status quo on which one it should be right now, but those
I
are
was going
two
to float
names.
out coralloides as well,
Mhm.
but I didn't actually know that bit about the confusion between the two, if they're independent or what's going on there.
I think they're the same species. It's just a matter of which name takes precedence. I'm pretty sure. So those are this. Another thing that you might consider. I don't think it's a Ramariopsis because
Oh
these
yeah.
these tips are really look flat and pointed, and
Right.
that's a Clavulina cristata or coralloides trait.
That Ramariopsis, it's like then the species name starts with K or something. Is that?
Quincei, I think, something like that. There's another one that's supposedly pretty bad, toxic mushroom that I don't know if I've ever seen it because it looks the same as Quincei. So that's kind of scary. But this thing here, which I think is Clavulina coralloides cristata, usually grows under conifers, but not always. It could be in other kinds of woods. It grows on the ground usually, or maybe from really, really decayed wood. Probably the
Yeah.
mycelium probably crawls up on it.
This right on like a very decayed white pine log. It was like right next to it, so
Yeah,
probably growing on uh, material from that log. You know what I mean? Yeah.
this looks like this picture right here really looks like Clavulina coralloides. Um, Ramariopsis quincei usually grows in beech woods. It's sort of a late summer, early fall species in my experience, and sometimes it turns a little bit pinkish, but it doesn't have the tips that get flat and pointed like this. The tips are a little more blunt. And I think there's a difference in spore morphology. I forget what the spores are shaped like for these guys.
Can you show my spores, Chris?
Oh, I didn't even see this other.
Yeah, I forget what they look like. I don't think they're elongated like Ramaria. Yeah, they're kind of short. Yep, there you go. Subglobose, broadly ellipsoid, like in
Yeah,
that sort of range. And I
they,
think Ramariopsis has spores that are almost round. I'd have to review that.
these spores look pretty similar to the Myco Quebec um, spore shot for coralloides. They have it as coralloides.
Cool.
Yeah, I think cristata might be a name that's not used now. I don't know. Both names seem to be pop up now here and there.
I mean, I don't know if it's useful at all, but maybe it's the outsides of the like the cell wall or something. But they have a real tomato seed look to them. Like,
I think the photo is just a tiny bit out of focus.
yeah.
Just a tiny bit. Yeah.
Just a tiny bit.
Yeah. Isn't that great?
Yeah, I know. When sometimes I'll take 30 shots of the same spore view before I get one that I'm even somewhat satisfied with.
Yeah, for sure.
And that shot right here looks very what I would call coralloides. But I don't know if I'd ever run into cristata uh, as a name before, but yeah, this one looks like that.
I think Mushroom Expert uses cristata, but a lot of times they're behind. They don't update their taxonomy enough.
Mm.
Cool, thank you. And I just have, I think maybe just or two more. So I'm thinking this is an anosogly. It was quite large, it's, maybe 4 inches across the cap, and really interesting look to it. And then it has sort of a hole in the center, which is interesting. And this was in mixed hardwoods, mostly oak, but I'm not sure which kind of oak. And then
Got spores. I think
yeah.
it's a Entoloma.
Oh, okay. I thought they were anosogly spores, but I'm still not sure I'm able to tell the difference. And
Yeah, they can be pretty similar. Inocebi spore morphology is all over the place. You know, like elongated ellipsoid, bean shaped,
Oh, look at them babies.
short ellipsoid. Um,
oh,
also-
thanks.
yeah, and
Oh,
sometimes
yeah.
Inocebi spores look like this. But you know what gives it away? The Entoloma spores. Look at the great big apiculus that
Yeah.
kind of juts out. You know, on the one end.
Oh, okay.
Yeah, you can see it on some of them. Some of them aren't profiled correctly to see that, but you can see a number of them that look like that.
What species Entoloma might it be? Any ideas?
Well, back to the Ramaria comment.
Okay.
I agree, Dave. Those are Entoloma spores.
Yeah, I think so.
But as for species, man, this thing is unique at least. So maybe you can get something, but
There's
I
a
never.
lot of Entolomas described on Michael Quebec. You know, if you want to put some time in and you know,
Yeah,
Yeah.
they have like 150 species on there with pictures and descriptions.
Nice. Okay, cool. Well, I mean, I sure would like to, of course. It's just finding the time. So yeah, thanks. I'll try. I'll see what I can do. Really appreciate it.
That thing's beautiful.
It was really neat. Yeah.
It is a cool one. You know, and that's why I'm saying it's such a unique growth form. You know, as long as it's not some one-off, right? Maybe you get some answers out of that.
it looks a little bit like strictius, but the stem is usually longer on those.
Yeah, do we have a stem, a shot of the stem? Does it have that strictius twisted thing going on?
Right, yeah. Yeah, that looks wrong. This is something else probably.
Yeah, I
Yeah,
agree.
this is
Yeah,
different species. Good find, you know,
it is
whatever
a
it
good
is.
find. Look at the gill edges, how they're a bit, you know, not quite jagged, but they're at least wavy.
For sure. Really wavy. Oh yeah, look at those. Right. And the margin too is really wavy, isn't it? Or would you call it that?
Yeah, and when I say edges, I do mean like the margin.
Oh.
Yeah, there's some action there. You know, they're not completely straight or smooth.
Yeah, I think Hunter's asking about the cap margin, the way it's like undulating a lot.
Oh
And
yeah, that
yeah,
also was, you know, a unique thing, right?
that's
Yeah.
interesting. Yeah, I mean, Strictius generally doesn't seem to do that. So yeah, it's an interesting one.
Okay, well, I'll look at Michael Quebec and see. And then I only have one more. I sent doubles. Jason
Ooh.
informed me that I sent this twice, and the other one I was gonna send, I'll just do another time. And this is like one of those polypores, at like what Fomitopsis maybe, or I don't know.
Mount CA, they call it now, I think.
Oh,
Was this on pine?
oh,
Oh, it's on oak. Oh, wow. It looks like the red bulb polypore, but I guess it's something different.
neat manifestation though. Look how it's like two
Yeah,
trees. I mean, I'm pretty sure.
yeah, it grew out. It looks like
It
it grew
grew
out of
out of
one on
this
the
one,
left.
maybe.
Yeah, into the one on the right and just
Right.
wrapped around it. Yeah, cool.
Whoa.
So Moonsei is the genus, or do you think something
No,
else?
Fomitopsis montsei is what the
Oh.
name for what used to be Fomitopsis pinicola, which is now recognized as strictly European or Eurasian. And but that grows on pine. So if this is on oak, it's something different.
Yeah, and it's not Robineae because it's not on locust.
Yeah, there's a few of these Phellinus type things that grow on oak. Might be one of
Yeah,
those.
that's what INET was pushing. Yeah, Phellinus, but I'm not sure which one. And no spores on that. So, but I thought I'd share, as my finds were a little weak this week, so what I got.
We all
Nah,
need to
that's fun
brush up on
stuff.
we all need to brush up on our polypores because we all find polypores and none of us know what to call them.
Yeah,
Well, I'm guessing that this one, this red margin is probably not a typical trait for whatever this is. So it's an unusual observation, I would say.
Really, I would just think that's the new growth, and that's what that new growth would look like.
Yeah, well, I would think it would be more creamy or white or not. It looks really pretty red, but it might just be something present in the wood, that's causing that.
Nice stuff.
Yeah, I think if you know where that is, you got a couple of years to figure out what it is. It'll be there for a while.
yeah, that's right. Okay, I'll keep working on it. Thanks for letting me share.
Thanks for sharing. You say, Dave.
Could we wait until 8 o'clock because I don't want to risk having the Jeopardy theme song get recorded. And
I forgot about that whole thing.
for those of you who don't know what I'm talking about, I was speaking on Taxonomy Tuesday one time, and my wife watches Jeopardy, and the theme song got recorded, and we think that's why it was removed from YouTube. So I'm going to try to be careful about that now. But anyway, somebody else go.
Does anybody want to go from their computer? I don't see anything in the chat. Was there Brandon and Douglas?
Okay, this is Doug. Can you all hear me?
Yeah, let me
Yeah, I just have two photos I sent you, Chris.
Are you in San Francisco?
No, I think it's just a backdrop. I'm not sure how I got that. I did live in the Bay Area when I was young. Maybe it somehow popped up on my image. I'm not. I'm outside Philadelphia.
Oh, cool.
I'm not sure I'm seeing the images. There are two. There should be an upper side and a lower side, Chris. Yeah.
We're seeing them.
Yeah, I'm not sure why I'm not, but at any rate, I was just walking in a little woods near my home, just a quarter mile away. couple days ago, and I saw this polypore on a downed branch of about an inch, inch and a half in diameter, and I thought, well, that's interesting. It looked a little bit different. It was kind of not hairy, but tomentose on the top, and on the underside, if you can zoom in, I hope the photograph's good enough. It had this kind of tooth-like, maze-like structure. If you play around there, you'll see it. It's not the best, but I thought it was, what they call Cerrena unicolor, mossy maze polypore, given all my choices. And I don't know if anyone wants to comment, if they've seen, I'm sure you've seen it. It's not that uncommon.
So this white part is the underside?
Yes. Yes, it is.
Yeah, I would say it's probably not Cerrena unicolor.
Not? What do you think it?
Um,
Go ahead, Dave.
um, I was going to suggest the underside didn't look like Cerrena unicolor, but the description provided does match Cerrena unicolor, the maze-like pores with elongated walls. I believe that's a Cerrena unicolor trait, so it might just be that the photo is not
Yeah,
accurately
both-
depicting.
yeah.
Well, I'm seeing kind of big pores on the underside.
Well, it was, yeah.
Yeah, I think Cerrena unicolor has pores that get fairly large. I'm going to look that up right now.
I mean, is it something that one of those that you look at the pore surface, and if you're not looking right on, it can appear more irregular and
Yeah, I suppose so.
I mean, there are some spots like over here that
Yeah, that
are
looks
definitely.
kind of looks slotted more than, perfectly cylindrical or circular, I'm saying.
Yeah. It seems like it's like really young or kind of not fully developed yet, too.
Or, if indeed this is Cerrena unicolor, potentially something has eaten those maze-like pores away, which I have seen before. But I would venture this is something else. I would say this is something else.
So Tramedes maybe, or
Maybe. But I mean, I guess, it's, mm, yeah, I think I would not call it Serena unicolor, but I don't know what it is, and maybe it is Serena unicolor. Could be wrong.
Yeah, does anybody have a good recommendation on polypore books?
Besides, the Beset's polypore book, I think is still pretty good.
All right.
Which I have. Bust it up and see what it says.
same. I need to look at it more.
I was going to say that all these polypores, regardless of what it is, can degrade lignin. A lot of them are white rot fungi. And Serena in particular has all these industrial applications now. People have cloned the gene for lactase, which is one of the copper phenol oxidases. It's a very old enzyme described 150 years ago, green colored. And they're using it now for bioremediation, detox, decolorization. And there's a lot of industrial applications, quite a bit of research on this particular uh, fungus, if it indeed is that. But they're also looking at other lignin degrading enzymes for industrial and medical uses, such as detoxification of aflatoxins. So it's just sort of interesting if you dive in there, because lignin is such a stable molecule. It's hard to degrade, but it evolved maybe 450 million years ago when vascular plants started to have a vascular system, and it solidified so you could have stems standing up. And it's thought that the rise of these lignin degrading enzymes occurred uh, at the end of the Carboniferous and sort of ended the coal deposition. And then there was this big flowering of there are many of these enzymes uh, that have been described. So it's just kind of interesting if you get into the biochemistry of lignin degradation. That's not to say that you don't need, with white rot, you also need hemicellulose degradation and cellulose degradation to fully degrade. But brown rot doesn't produce these lignin degrading enzymes. So it's kind of an interesting thing to think about when you look at these polypores that degrade lignin. So yeah, that's pretty much all I had to say about it. I just find the biochemical aspects of it very interesting, just to ponder and think about how they evolved and how they play a big role in degrading wood.
That is so fascinating, and it's so interesting to think that that can be utilized outside of making paper or whatever, in so many applications, like you mentioned. I'm interested in learning more about that. So
Yeah, if you just look up lignin degrading enzymes, you'll find a whole literature. They are used in the pulp industry already, for decolorizing and degrading residues in paper manufacture, already being used, these particular enzymes. And there's a big interest in scaling up industrially one type of enzyme is called a lacase, uh, which Cerrena unicolor makes in a huge quantity compared to other polypores. And they've actually cloned it and expressed it heterologously in Aspergillosis niger so that they could produce big quantities of it. So if you take a look at lignin degrading enzymes, lacases and heme peroxidases, Hunter, you'll see a very interesting literature there, and makes you realize it's a big world there. It's not just, it's not just, oh, I saw this polypore, but wow, here's all these things going on with it.
yeah, the function of the rotters, I mean, just, it's so interesting. The brown rots have their role and the white rots have their role, and but then it sounds like, wow, it can really be expanded to so many things. Yeah, I can't wait to look. Thanks, Doug.
So is the lignin degradation a necessary ingredient in reducing wood to soil eventually?
Yes, it is, Dave. In plants to produce lignin, and these white rotters can go all the way to CO2 and water, not just soil, but they require other assistant enzymes, not just the lacase. You have to have manganese peroxidases. There's a whole list of things. It's really complicated. There are many different types of lignin, so that softwood lignins are very different than hardwood lignins, as opposed to conifers. So the chemistry of these subunits, they're built on phenylpropanoid units, is different in these three classes of broad classes of trees. So yeah, I mean, like you said, Dave, you could go to soil, I suspect, or which would be kind of a partial degradation. Fungi are the most active, but bacteria also contain some of these enzymes, and they've been around for a real long time.
It's a weird thing. There was a tree on my way home from work, and every year it got uh 2 or sometimes even 3 runs of sulfurous going up it. And right towards the end, that last 2 years, it would fill the whole dead tree top to bottom. Uh, and when that thing finally came down and the way that it fell, it kind of hit the sidewalk, so no one cleaned it right up. I noticed it was down and I pulled over, and you could just pick up a chunk and just squeeze it in your hand and it would instantly turn into this like almost like almost like damp peat moss.
Yep, that's I've experienced that. It's very, very interesting, very cool. Makes you realize that if we didn't have these fungi, we'd be buried a couple of miles in tree trunks by now.
We wouldn't be here.
Yeah,
Yeah.
exactly.
And it's a fascinating discussion. Just to be clear, this is not Cerrena unicolor. I'm
No.
pretty sure of that.
Yeah.
Are you certain of that, Brandon? What is it
Yeah,
specifically?
I don't see a single tooth or elongated pore.
Yeah,
on the
I
other
think.
side
Well, I
of this
think they
thing.
might just be. It might just be young fruit bodies that haven't expanded enough yet. That Doug said that the pores were maze-like, and that is a Cerrena unicolor trait. And the descriptions say that they start out white. So these being white would be young ones, and perhaps you know the tubes have not yet elongated to be able to take on that tooth-like uh, appearance.
Yeah, it's a little bit my fault. I have an old iPhone with not the greatest camera. I looked at it with a hand lens, and they were more maze-like. They weren't really rounded. So this is not the best photo, and it's out of focus in some areas.
Yeah, the other thing that's said about Cerrena unicolor is the size of the pores varies quite a bit. 1 to 4 per millimeter. 4 per millimeter would be fairly small. Most of the photos you see of Cerrena unicolor show pretty mature fruit bodies, and the pores are pretty big by then. So that would be the ones that are like 1 per millimeter. So I don't know. I wouldn't rule it out.
1 per millimeter is not small.
No, but that's the upper end. So 4 per millimeter is the lower end given on the size of the pores, and that's
Right.
actually pretty small.
Yeah, that's fair.
Mm-hmm.
Well, thank you, Douglas.
Oh, you're welcome. Thank you for letting me talk about that. I'm going to hop off.
Well, then have a good evening as well.
No, I'm going to stay, but I'm going to mute myself and take away my image. Here we go.
Oh, that one's mine.
That one is yours.
All right.
Brandon, I'm
yeah.
sorry to interrupt. Before you start, I have a question to ask of anyone. Can anybody check Mushroom Observer and see if the proposed name button works for them? Because it's not working for me, and I use 2 different computers. Both are Chromebooks, so it's Google Chrome, and it's completely dead. And I just want to see if other people can reproduce this. I'm just curious.
Oh, so
Thank you.
I'm sorry, Igor. What you want to see is if someone is able to propose an identification.
Yeah, open observation and click on the proposed name button and see if actually the window pops out.
I'll do that now.
Okay, thank you.
Yeah, cool. maybe it seems like Zoom was a little bit messed up. Maybe there's something going on MO as well.
Well, I mean, I
Um,
report a number of bugs to them in prior days, so they're doing something there. So maybe something went wrong. It's possible.
I just tried on my iPad and it just like highlighted it and didn't open it.
Well, mine doesn't even highlight, but I get like a little link on the bottom of my screen, like a very long link. So yeah, it's not working. All right. Thank you. So
yeah,
and you were Hunter. You were working off what device?
iPad,
What's that?
iPad.
iPad. Okay. And what browser are you using?
Oh, Chrome.
Chrome as well. Okay. All right, Brandon. Sorry about that. Go ahead.
no problem. This is a good one we can, I think, breeze through. Found this at the club's foray this past weekend. It was on decayed conifer wood. I think it checks all the boxes for Tricholomopsis decora. Yeah, the most prominent feature are those kind of scales on the cap. Yeah.
Cool.
Yeah,
Oh, wow.
I don't see it all that much. I don't think it's too uncommon. You just have to be around well-decayed conifers. Yeah, it's a little
That is
beat
some
up.
intense cross veining.
Well, no, it's a little beat up. Maybe you can't really see that.
Eh?
Yeah, there is some cross veining going on. But there's also some bacteria moving in on this thing, so it's a little bit old. Can't really make it out all that great.
Yeah, so it's a pretty common species around here on white pine wood. Usually late, midsummer on into the fall. And the only thing I can think of that you might confuse it with would be a Gymnopolis,
Hmm.
and then you can tell just from the spore color.
Yeah, I actually thought, because I found some more mature ones that also probably had like a bacterial infection of some kind, that uh, those scales weren't as noticeable. And when I saw those, I actually thought they were Gymnopolis. But uh, when you can find one in decent shape with the decoration on the cap, the scales, I think that can kind of let you know what you have there.
Yeah, this looks like Tricholomopsis to me. And look at how the scales kind of, when you radially move away from the center, um, they kind of become a little bit streaky, sort of.
Yep. And then the last photo in this observation, because I feel pretty good about calling this Tricholomopsis decora. Um, yeah, I considered the what is it?
Sulfurioides, I think it
Yeah,
is. Yeah,
I consider
that one doesn't have as many scales.
right. That's right. That's what I saw.
Mhm.
But this last photo is going to be the observation for the next photo. I found these growing, just down the log from it, and I actually I think they're also Tricholomopsis, and I think they might be young Tricholomopsis decora. But they look so different. I mean, they're so dark. So if we can go to that next observation.
That was going to be the first thing I was going to ask if you thought those were the same thing, because they might be.
Yeah, and I'm not sure. But I think they might be. And if you
It
can
looks like
zoom
they
in,
may have dried out in situ before they were able to mature.
mm, that yeah, I hadn't considered that. Most Yeah, that's actually probably what happened. Maybe got burned up a little bit too.
Yeah, yeah, in the heat. And they probably are Tricholomopsis. It might be a different species, but it's probably the same thing.
That's that's
It's an
pretty much
anomaly.
that's pretty much where my brain landed. Maybe it's a different species, but more likely it's just, a young, didn't wasn't able to fully mature decora type thing. There is this Tricholomopsis scabra, because I didn't do the micro on this, but I did kind of look into some darker Tricholomopsis, and there is this Tricholomopsis scabra that doesn't look dissimilar from this, but I also can't really get a good description of what scabra is. So, yeah, it, probably Tricholomopsis, probably the same thing that we just saw, but thought it was neat that they were darker.
Interesting.
Yeah, there's also rutilans, but that's usually got more sort of red
Mm-hmm.
color
Right.
on it. And that one I usually find not right on wood like this, but like on the ground from wood.
Same.
Yeah, and it's got kind of more of a, I don't know, handsome stature to it, if that makes any sense.
Yeah, they look really nice. They have those nice yellow gills,
Yeah.
and the cap is usually a little bit reddish. But the thing that reminds me is the really, really scaly cap on these.
Yeah, and I have them. Like I said, I haven't gotten to the micro on them, but my present thought is they're most likely the same thing as the decora that we just looked at. But we can move on.
I mean, like, if they didn't dry out, would they have been a more yellower?
Well, that's my question. I'm not sure.
Yeah, that's what we're sort of presuming. Oh,
Yeah,
how big
yeah.
were those caps? Those dark ones?
Um, quarter size. You know.
Quarter size. So that would be small
Yeah,
for
yeah.
Tricholomopsis decora, or decorum. I forget which way it goes, but so
Yeah,
so that would
probably
make sense then that they're just
about
not expanded enough
yep,
for the
about half the
caps.
size. About half the size of the yellow one we just
Yeah,
looked at.
yeah. Hmm.
Hmm. Yeah, because
So
even in this, they look like they could take on that hue a little bit.
right, right.
That's cool.
The way these are growing, I know we're thinking it's Tricholomopsis, but the Gymnopolis would grow very similarly, right?
Oh yeah, yep.
Yeah.
Yep.
That's a cool shot. What is that?
All right, so this is a slime mold, also from the. I didn't collect this one actually. This was growing on Tremella mesenterica, witch's butter, on the ID table, and someone was like, "Hey, there's a slime mold on this." Um, so I'm calling it Physarum or Physarum viride. Physarum viride. Um, you can see got a beautiful yellow.
Ooh, that was perfect.
Yeah, look at that one. And doing the typical Physarum, uh, Physarum. I can never say that one right. Kind of cracking with the peridium there.
There we go. It's comically small.
Yeah, they are very small. But
Wow.
I feel the viride, Physarum viride, I would label that as promising. There's a couple other Physarum species that it probably could be that when I get to the micro, I think I can I think I understand what I'm looking for. I think it's all about spore size, which is nice. You know, are they really small or are they over 10 microns, which would be a viride thing. Um, but yeah, were you guys able to see that they're on a little stalk, like a little foot? I don't know if we highlighted that at all.
Not really.
Mhm.
Yeah, there you can see that foot.
That's one on the end there, and then
Ah,
Oh, that one that's like
this one.
is fallen over a little bit
Mhm.
on the left.
And then the one in the dead center, he's highlighting the.
Oh, and then that one too. Yes, I
yeah.
see it.
So
Uh,
I saw Virginia was here earlier. Is she still here?
How do I do that?
No, I just checked. She's not here. But yeah. Promising ID for Physarum viride.
Nice, nice photos.
Cool.
I'm not
Thank
gonna
you.
be with you.
Man,
All right, and then this thing I found today, and I feel like we saw this here before. I think Lila might have shared it. And I hope you guys can make it out from this photo that this is some kind of like crust fungi that is raised up. I mean, it's like a crust fungi on stilts. It's like 5 millimeters raised.
it's like volcano.
Yeah.
What part is the fertile surface? Can you tell? Is it the brown part on top or the white part?
I don't know. I don't know the answer to that. That was the first. I like I said, I found this today, and when I cut it open, first of all, it's really, really tough to cut through, like leathery. And I thought maybe this thing was an asco, because of that, and that fertile surface would be the brown part in the center. But these things are actually in order Russelales. This mushroom is related to a Russula.
Huh. You know what
Yeah.
you can check then? If you can find some spore, oh, you would need Meltzers though, or Lugol's, something like that.
Yeah.
Yeah,
Amyloid it
yeah,
is.
you can see if the spores have amyloid warts.
Hey Dave, could you pronounce that second one that's not Meltzer's again?
Lugol, I think
Lugol.
it's Lugol's.
That was the one we were talking about on.
Yeah, no, I know. I was hearing a difference.
Between the
Oh,
lip and
okay.
out.
I'm not sure what you heard, but it sounds like maybe
Maybe I was mispronouncing it or throwing some Jersey accent stank on it.
Listen, Nina just hooked me up with some Meltzer, so I don't need to
oh,
know it
great.
that bad. It's just interesting because the both of you went and pronounced it differently immediately.
All right. But yeah, was it Lila that shared this thing with us maybe a couple months ago, or am I completely making that up?
I don't know, but it is super cool, whatever it is.
Yeah,
Yeah,
I
it's
think I remember first sharing it, but maybe is that just like a pencil? Oh, there. Oh, it's bigger than I thought. That branch. Okay.
yeah. I think this photo does the best at like showing you how these kind of cylindrical fruit bodies. I mean, they do, or they're pretty much perfectly cylindrical, and then they grow into each other. Um, that's what I read about it. Is it Gypho to something?
And is this something like this stick was laying in this orientation?
Yes. Yep. This is the in situ photo. This is how I found it.
So I wonder, like, if it's, did this stick look like it was laying there for a while? and this thing grew this way, or could it have like fallen out
No.
of the tree this way?
Well, I mean, there's always that possibility. But it's growing, you know, vertically, certainly. Where the place that I go, no one goes, so no one is. This stick has been undisturbed for a long time.
Cool.
And this is one of those things that has two growth modes. You know, it's got like the other growth form for this thing is just a white resupinate polypore that looks like 1,000 other white resupinate polypores, and then it has this growth mode that is pretty unique. So I think I have the ID correct, and it doesn't seem like a super common thing. So
The ID is Skype
yeah, you got to go to it. I have to read it to
yeah, for sure.
Scytinostroma artocreas.
Cool.
Scytinostroma artocreas.
And it's in Russelaceae.
Yeah,
Yeah. Okay.
Russellales.
Oh.
So the higher order
Okay.
over the family.
Yeah. Okay. Okay, cool. Thank you. Neat. So neat.
Yeah, thank you guys.
Is it?
I think so.
Does anybody have anything from their computers?
I can try sharing from my phone.
Go for it.
Bianca, hi.
Oh, friend.
Something
So
you
I'm
just
so
found
sad
on
I
a
didn't get
walk?
a chance. Sorry?
Something you just found on a walk?
Well, not exactly. It was semi-planned. I was so sad I didn't get to join you at Shark River this weekend. I had other agenda to do, but I went yesterday to a different section of it. It was a River's Edge trail. Let me see if I can share this. Oh boy. Might be easier just to share my screen and see if it works. Photos. No, it's not letting me do that. If somebody else has something first, they are welcome to attempt to share. Wait, maybe this will work. Okay, so can you see what I see or no? Not yet.
Not yet.
All right. Well, I found many different mushrooms, but I had specific ones I wanted to share. This will be 3 in 1.
Oh, good.
Oh, there we go.
All right, so those 3. The one on the right is a hedgehog mushroom, and the other 2 are boletes. I think that orange capped one is the that orange was so vibrant. It looked like a traffic cone. And the middle one, it actually stained blue, where you see the X is in the pore surface, and then it faded. Let me see if I can show you the tops next.
Do you have a name for the one on the left? The orange cone, the traffic cone orange one?
I had a name, I believe it may have been, but give me a second to show the other one. Oops, that's from photo. This guy. Where are the tops? Here are the tops. Those are the 3 tops. So the hedgehog one is now the bottom. And yes, and that top one is also that current one that's orange. It's stained over time. It turned brownish. within a couple hours. I can show that as well. Do I have a name for it? So the name I thought it might have been, let's see if I can find it. This guy, Tylopilus balloui.
Yeah,
Yep.
I'd say so.
Right. Sorry, I'm very excited. I was
Yeah,
able to
no,
get
that's a good idea, and that color orange, you're right. I've seen that in the woods from a mile away. It's like, whoa.
Yeah.
There's Tylopilus balloui from 100 yards, right? My concern about the thing you're calling a hedgehog.
Sarcodon, is it?
Yeah, I think that's probably a Sarcodon impicutus, I think is the species name on it.
It's it's a Sarcodon, or at least what used to be called a Sarcodon. I think some of those may have been moved. I'm not sure. Like 2 years ago. Let me look on Michael Quebec.
To Burkhandera or something. Not Burkhandera. Bankandera?
No, those have white spores, I think. I think they moved a few of them to Hydnellum, actually.
Oh, okay. So
I'm not sure. You know, I might be wrong about that. I'm gonna check, see what's on Michael Quebec.
The balloui, is that an edible, good edible?
I don't think it's considered to be very good.
No, okay.
That's
Is
good to
there
know.
another balloui on bolete? Oh, there's the underside. Nice.
Yeah.
So it looks
that's
like it has
interesting.
very small spines.
Yeah. It was really cool to see. I was shocked that it actually has spines instead of either pores or gills.
If the bottom of the stipe has a greenish bluish tinge to it, then I think it can be identified to imbricatum or something like that. I forget. I'm not really knowledgeable about these sarcodons or phelidons. Now the one in the middle is bicolor. That's Byrangea bicolor.
Oh, it's my first time finding it. Thank you.
Yeah,
I
pretty
wasn't
sure
sure
that.
because it looked like it was not as, I thought the difference between the top and the bottom of the stipe would be more of a difference, but guess not.
Yeah, the other photo had all the characteristics of Byrangea bicolor. This one, was sort of still questionable. I mean, I knew it was a bicolor-like bolete, but it could have been a Lanmao instead. But that photograph, the colors, and the pigmentation on the stipe in the kind of a blotchy, spotty matter, I think that's a trait of bicolor. What do you think, Dave? Bicolor?
Oh, I was looking at Hydnellums. I don't know. I honestly.
Can we zoom in, please?
It looks like it's not a real dark stain on the.
Yeah, it's bicolor. Yeah.
Yeah, it just seems a little bit dark on the stipe, but I'm not going to disagree with you there.
Yeah.
It's bicolor.
When you cut that thing, it shouldn't stain at all inside. And
the stipe, it should not stain when I cut the stipe.
Yeah, when you cut the mushroom longitudinally in half, you should not see any staining on the flesh on exposed context.
Yeah, but it might darken, and actually might darken considerably when you rub or handle the stipe.
Yeah, I mean the surface will blue probably, but the context inside usually doesn't blue.
Right.
Yeah, I was referring to the flesh, not
right.
The surface.
Yeah, I just wanted to make sure we were clear.
Yeah.
On the two different potential types of staining. Oh, look at the Sarcodon. Look at, there is a little. You can see the blue on the bottom.
Yep, that's the one.
Yeah.
Any more information about this one? Is it's a big enough mushroom? Can I cook it, or is it not edible?
Probably bitter.
The Sarcodon is probably bitter. Yep.
Yeah, not good.
And are bicolors normally this small? I was expecting them to be bigger.
It varies. Sometimes they're small. They're not typically a large mushroom, maybe medium-sized. But it varies. But the ones I've seen are typically modestly sized.
Gotcha.
There's a
Sorry.
Sarcodon that they collect to eat in the Rocky Mountains. It's probably something we don't have here. In my experience, all the Sarcodons that I've ever found were bitter. I would just taste them a little bit to see, and they were always
Oh.
bitter.
Gotcha. So this guy had a funky smell, obviously with latex, but it's probably the biggest latex producing mushroom I've found personally. And when you're ready, I can change it to a different view.
Is that Phellinus, you think?
That's Phellinus.
Yeah.
Oh
Yeah.
yeah.
I eat those. I like them.
You like finding them?
Yeah, I like finding them, and I ate them too. And I think they're a beautiful mushroom.
Oh,
Beautiful.
it's stinky. I don't know if I could eat this one.
It's funny because the ones we have here, I was saying, have this sort of they're supposed to smell fishy. But mine smell to me really good, like kind of like a sweet curry or something. And so I know Dave said it might be a complex, and it probably is. But I think it's a beautiful mushroom. But
How do you cook it?
They do. I haven't done anything real special with it yet, but just saute and eat it. That's it.
Gotcha. Well, I definitely saw and took home some chicken of the woods, and I wanted to share actually my method of preparing it. It really worked out very well. So this is the chicken after it was prepared. I actually, before I battered and fried it, I soaked it in chicken stock for it to have a juiciness to it. And then flour, egg, and panko breadcrumbs. And it fried up very well in avocado oil. Just super nice. I've been sharing it at work.
Man, that's fancy.
Thank you.
I'm sorry. Did we get a name for that Sarcodon?
No, I'm gonna look again. Yeah, I got it.
It's the thing
Well,
you thought it. It's the thing you thought it was.
It could be Hydnellum scabrosum. At least in the treatment of Kuo, because for that one the stipe on the bottom turns that slate color. So Sarcodon scabrosus was the previous name, and they moved it to Hydnellum. There was a paper a few years ago on the phylogeny of these dark spored toothed fungi, and a lot of Sarcodons have left the genus and jumped the ship and became Hydnellum.
Okay, that's what I thought. I thought I remembered that.
Yeah, it's a paper
So
by some Norwegians or Danish people. I forget their names, but it came from Europe.
And I found a ton more, but I will step aside and let somebody else share. And maybe if there's time, I can share more at the end.
No, show your thing.
Yeah, let's see what else we have.
Okay, all right. I will go through a couple. This one was super fancy. It was dripping even though it hadn't rained for a while.
Oh.
Oh yeah. Okay, spraguei.
Yeah, it's
yeah,
our buddy.
Fomitopsis spraguei. Yeah.
Yeah. There's a lot of those this year. It seems to be a good year for those.
Yeah.
I've seen a lot of them online also. People are posting them.
Yep. And we see this like on almost every foray, at least one specimen.
It's spraguei is the species, but what's the genus?
I think it's Fomitopsis now.
Fomitopsis, it is, yeah.
Oh, okay.
Yeah, on the older sources are going to call it, I think, Niveoporofomes or something.
Porofomes, yeah.
Okay. Fomitopsis spraguei.
Yep.
Thanks. Glad you showed that.
Thank you. So this one almost looked like a little flower with the nub at the top. Let me see if I can-
Entoloma.
Yes.
Yeah, it's an
Entoloma.
Entoloma. So if we can see the stalk, we might be able to see if it looks like it's strictius, because that could be strictius.
Isabellina.
Isabellina or Isabelinum. I can never get these endings straightened out.
He has a left twist or a right twist. Let's see.
Yeah, it doesn't twist, but you can see the streaks. Right?
Mhm.
Yeah, I think that's what that is.
Which one, Dave?
Strictius variety Isabellinum, is it? Or is it I think it's Isabellinum. Or yeah, I think.
I
I'll
think
check.
it's Isabellina.
No.
Oh, it ends in A? Okay. I can never get
Let's
that.
take a look. I'll check the name.
Yeah, so that would be Entoloma strictius variation Isabellina or Isabellinum that they're checking on.
Isabellinum is the correct spelling.
Nice.
Oh, okay.
So this almost looked like a white mushroom like you would get in the store. And even
Yeah, it
more
won't
so-
taste like it though.
Oh, I'm sure it would not. And here's the top.
Ay, ay, ay.
Yeah, it's a Lactophyllus deceptivus, probably a young
Yep.
one.
And then here's the split.
Oh, yeah.
Wow.
Look
Yeah.
at that incurved. Yeah. Yeah.
Yep.
Nice.
And the depressed center that's a little bit brown. They get to be scaly when they're older because the cap surface breaks apart a little bit.
Yeah, it looks like a parchment, inside.
Yeah.
The surface very rough and scaly and brown. And of
Mhm.
course, that twist, of the cap margin. Ionic column, as I call them. Yeah, almost like that scroll-like thing in the ancient columns. So you see, you get
Mhm.
Doric, Ionic, and Corinthian. Well, this is the middle one.
Very nice. All right. I feel like this is a pop quiz for you guys. You're enjoying it too much.
Too much, yeah.
All right, let's see. Let me see if I can stump you with this one. This is how it was growing. A little bit of the edge into the soil. Then
That was soil. That was the ground.
that was it was a slope. So it was like earth that had fallen away and it was growing up along this slope.
It's growing sideways from a vertical
Not
cliff.
sideways. It was growing vertically, but it was just so much along it that when the umbrella opened, it grew into the dirt a little.
Yeah,
So
well, we know what this is.
You know what this is. Is it
Lepiota.
No,
Amanita onusta?
that's right.
Oh,
Amanita for sure. I knew that much.
Okay, yeah. Now, once you see the bottom, right?
Well, the top was the tail.
Well, there are lepiotas that look like that, and also echinoderma, but that usually grows on woody litter.
Yeah, but the vulva is gray, and its
Yeah, the
disposition
color.
is also
Yeah, okay. Yeah. But it doesn't. There are some lepiotas, and Leucoagaricus probably that look a little bit like this. But yeah, once you see the bottom, that's none of those have bulbs on the stipe base.
Yeah, and you should see the same wartiness, gray warts on the bulb, at least on top of it.
What kind of
Unless,
tree did you find this by? I'm interested because I usually find it by either hemlock or birch.
It actually didn't seem to be. It might have been a root that it was growing from because there was no particular tree right next to it. There was just that soil.
Yeah, well, it wouldn't grow from a root specifically. It's a
Mhm.
it's a mycorrhizal species that
Mhm.
but it will be in the vicinity of some sort of tree, that the fungus is associated with.
Probably oak. If this is Shark River, it's a pine oak forest, and the species is, I think, mostly oak associated.
Oh, I find it with hemlock and birch.
Maybe it goes both ways. It kind
Yeah,
of like
yeah.
spines
A lot
and
of the amanitas do.
Well, she said she was there. Was that also on that River's Edge trail?
Correct. All of these have been from the River's Edge Trail.
Yeah, there's a bunch of birch along that trail.
And we did
Those
find
actually
a
all
noosta at the foray on Saturday. So somebody brought in a young anusta.
What do you got there?
So this was
Like
actually
a
a larger
video
one than I've seen before of this
Called
type.
Tritium.
It's a Phellinus.
Oh,
And then
oh,
here's the
okay.
other
I
side.
think it's a Phellinus. I could be wrong. Maybe it's a
Let's
polypore.
see the bottom.
Again, a
Probably
derma.
polypore. It's a polypore. I didn't recognize the stump, but once I saw the stump, I knew I was wrong.
Oh,
Say
so
Ganoderma, Brandon?
Not a Ganoderma, I don't think. I don't
no. How about Brasidolia? Or Brasidola? I forget if there's an extra I in there. So there's Brasidolia, Craterellus, I think.
Well, isn't that stiped? This one doesn't have a stipe. It's a thick polypore.
Okay.
It's a vase shaped.
I'm not sure that one really has what you'd call a stipe. Let me look it up.
Looks like a Ganoderma actually from the top.
It does. You're right. It does.
Yeah, I'm sticking with
Oh
Ganoderma.
yeah. If the pores are
think
really,
so?
really small, then it's Ganoderma, and it's not these other things that I'm mentioning.
Oh.
Yeah. So you would.
Ganoderma megaloma, something like that.
It's either that
Yeah,
one.
really, really small pores, and it looks like it has really small pores. Yeah, so I agree. That's probably a Ganoderma.
Oh, wow.
Yeah, it could be the megaloma or the. I mean, lobatum is a misnomer, right? It does sometimes form this vase shaped thing, even though most of the time that you think about it, it's just a shell fungus. Lobatum's not out of the question.
Hmm.
I think.
Well, that one's got a lot of room, so it's just all shelf, I think.
Not gonna
Yeah,
argue with that.
do we know the substrate? Lobatum is a hardwood thing.
That sounds right. I don't think there was a lot of hardwoods, different kinds of hardwoods there, but
That
this
thing's
was,
pretty
I don't think this
crumbly.
was a
So is
pine.
megaloma, I think.
Yep. Yeah.
Probably an oak tree, maybe maple. If there's maple there.
There's definitely maple there. 100% definitely maple there.
All right.
I like that picture. That's cool.
Thank you. All right. Let me see if I can do maybe one more.
The last chance to stump us.
It is a fun game.
I'm gonna have to tell my wife we should have went down that trail.
It is nice. Very, very pleasant one.
Check for ticks.
I did have a nymph tick on me, just the single one.
There you go.
It's probably a larva. There are no nymphs right now. I picked up one too, but I only found it crawling on me the other day on Sunday. So I'm not even sure like where it came from. Oh. Okay.
Well,
Yeah,
if
this
you're
is gonna
gonna
stump
stump,
everyone.
yeah, if you're gonna stump us, you gotta give something that has a name, probably.
Yeah, this one is a sequencing candidate.
No. Oh, darn. Okay.
Yeah,
Well,
Right.
let's see the bottom. Let's see if she dug it up.
It's an Amanita section virginata, most likely.
Yeah, well, that's what I would think too. But if it's dug up and we see a bulb, then it might be brunescens.
Well, Rattau has described this brown virginata that he called Shark River Ensis.
Oh, okay. That
Oh, so maybe that's the
Really?
one.
Really? I didn't know about that. Shark River Ensis, huh?
Shark River Ensis. And I think I found it somewhere else and I sequenced it, and that's how I found out about Shark River Ensis.
See, I thought it was going to be too young to be able to identify, so I was going to come back tomorrow.
Doesn't matter.
Do you know
Doesn't matter. Now I
if the
know.
cap has grooves on the margin, striations?
Here
Yeah,
we
it
go.
does. Okay,
There it
so
is.
that's what Igor says.
Yeah.
So I found my local mushroom. Very good.
Yeah. So there's a chance it might be Shark River Ensis after all.
Very good. Okay, that's enough for me. Thank you so much, guys.
Thank you.
Yeah, thanks, Bianca.
Was there rain there recently? Because it looks like you found some nice fresh mushrooms.
A couple of days ago, it was quite a storm. Yes.
Okay, there you go. Yeah.
Hey, Emily, you want to go?
Sure. I was gonna go anyway tonight, and then Igor has already posted on my mushroom and proved me wrong, and I'd like an explanation. So let me see if I can get myself sharing.
You get some explaining to do.
Called out in real time. That's
See,
good.
I haven't shared from my new computer yet, so give me a minute. Wow, open. Oh shoot, I think I'm gonna need to. I think someone else will have to do it for me. I think I need to change some settings and then sign out of Zoom. So instead of doing all that, I'm just gonna put it
Yeah,
in
put it in the chat.
Oh, they're from INET actually, not MO.
Well, it's on both. It's
yeah,
on,
you imported. Yeah, I think I saw that. Yeah.
it's just easier to get to the photos, I feel like, on INET.
Man, how did I do that?
Mm,
So another tooth
No,
thing.
no, that's not.
That's
That's not
Chris's actually. That's Chris's observation.
No.
Oh,
From
I just opened.
from a gazillion years ago.
You could just open the link in the chat and get another window and see. But let's look at what this is. We've got something up
Emily.
here right now.
July 26, 2023. Oh, this one is a Phellinus probably. Sorry, Hydnellum. Yeah, Hydnellum.
Who's sharing right now?
Yeah, Chris, you with us, bud?
Yeah, I had the one from the
Did you click on the link that
what's on the link?
Emily?
It says Emily Rollins.
We saw something else.
Yeah, you should stop sharing and start sharing again, probably.
Huh. Let me click the link. Is it still
We're not seeing anything.
hold on one sec. Where does it go when it shrinks? I
To be
don't
fair,
understand. Hold on. You're seeing like an email page right now.
Uh-huh.
Yep.
Uh-huh.
If I go to the chat, bitter bolete.
It is. Well, it's apparently not, but yes.
Okay, nice.
You guys see it now?
Yeah,
Yep.
we can
Yeah.
see it now.
Yeah, see it.
Man, that was weird.
So
Take one.
I was perfectly happy with what I was calling this until I saw Igor's comment on Mushroom Observer. And Igor thinks this is Boletus venacipes. And my concern with that is that it's not gray. And I thought that that had to have a grayish cap. And I had looked at that, but I'd ruled it out. So what am I not seeing?
Well, they can be that color, and I have an observation from a long time ago that has exactly that color.
Okay. I didn't realize they
So,
were completely brown like that.
Can we see the sectioned mushroom again? Because I agree with Igor. Yeah,
it's
yeah,
not bitter.
what about the
And
bottom of this inside of the stem?
yeah, Phellinus is never going to have that coloration inside this type to begin with.
Yeah, usually the it's venacipes, right?
Vinacipes, yeah.
Yeah, usually that one's got a little bit of like purplish stain inside the bottom of this type, but not always.
Not
But I think
always.
that's what this is.
Yeah, outside you saw the purple stains and then mixing with the yellow.
Okay. Yeah,
And of
yeah,
course,
a little bit of yellow. Yep, yeah.
As I call it, the aerodynamic reticulation, kind of thinner and very polished, separates that from greaseous. The pore surface is grayish and will stay so. Will never turn pink, as far as I know. Maybe brown, but not pink. And yeah, that's it's a pine associate, just like Phellinus, but it's a fairly common species in the pine barrens.
Yeah, I find that around here quite a bit. I found a few of them a couple of weeks ago. I found a few of them here.
Yeah, and that's how you separate it from griseus or greaseous.
Yeah.
Reticulation. The stains are not vinaceous, and the
Habitat.
yeah, and the caps are more brown and gray.
Yeah, caps are. Yeah. Griseus has a really gray cap that might fade.
And very, very shaggy, coarse reticulation. The reticulation on
Very
Boletus
coarse,
is
yeah.
always more refined and smaller meshed. And
Here's
never
an
shaggy like
interesting
that.
thing about this species. The types collection comes from the Dominican Republic, I believe.
Correct. And our hypothesis is that it crawled all the way down because we find it all the way into Canada, and somebody exported that thing with a pine tree. And because I don't think it would be a native species for the Dominican Republic.
No, it probably got imported on trees.
Yep. So again, you can separate griseus and vinacipes just by the habitat. This is a strictly conifer associate, whereas the other one is strictly hardwood.
All right. When I'd looked it up, there actually don't seem to be very many people posting, at least on iNat, with the name that Igor had proposed. So I will definitely change it then, so that way people know that this is a possibility.
It's mostly a coastal plain species, but we do find this farther inland. Dave finds it in his neck of the woods. We find it in Stokes. We found it.
Yeah, I find it in lots of places here, really. Wherever there's pines, white pines.
Yeah, I think Jared Albrecht found this in Montalto.
Yeah, I believe there was some there. Yeah.
Yeah, so
that's pretty far inland. Yeah, there's a little bit of the vinaceous staining on the outside of the stipe,
as well as yellow, and Phellinus
as
would
well
not
as yellow. Yeah,
like that.
yeah, it's what it is. So this used to be a variety of Griseus, I believe.
No. I mean,
well, for a while it was sort of proposed.
It was proposed for as a species from Japan or Asia.
Oh.
And variety fuscus of something.
Oh, okay. Yeah. That's right.
And,
yeah.
Right. And then when we sequenced this, we realized that, the type collection was sequenced much later after it was published. So the publication dates back to 2007, that paper. by Baroni and Ortiz Santana. But the sequences came back only like 10 or 12 years after that. So for a while there was a mystery. Somebody actually wanted to publish the species, and then they came across the sequences. So yep, now you know what it is.
All right. Thank you.
I heard you guys mention, I think, 4 species of red boletus. How many species do we have in northeastern U.S.?
Well,
Or in
Griseus, Vinacipes, those are the, I grouped them together because they are gray-brown.
Mhm.
Ornatopes, but that's a species cluster. There's at least 2 of them.
Oh,
And
okay.
there's also something that they describe in the Ornatopes clade. I think Berkeley described it. Curtis and Berkeley in the 19th century. Something from North Carolina, Boletus. I have to look it up. Not Cespitosus. I have to look it up. So probably I would say in total 5 species. Maybe I'm forgetting about something else. Maybe half a dozen, up to half a dozen.
And did you mention something called red boletus fumosus?
No, I said
Fumosus.
Vinacipes. Before we found out what it was, we used to call it Vertiboletus something variety fuscus,
Oh,
which is
fusca. Oh, okay.
from Japan.
Okay. Gotcha. Thank you.
So yeah, it's a small genus in North America, and it sits in the family subfamily of Lexinoideae, so it's a relative of Lexinum. Strange.
Nice. Is anybody else?
Well, Emily had 2 more, I think.
Oh, all right.
I did. Well, sure, if
Well,
you'd like to
Well,
see them.
well,
go
yeah. I mean, I
ahead.
saw them on the Mo. I thought you would be showing them
Okay,
as well.
okay.
Yeah, he's like, yeah, show those 2 other things. I don't know what they are, but yeah, show 'em.
They're not boletes. They're gilt mushrooms.
Yes, that's all right. I'll bring up the ammo. I'll put it in the chat. Just a second. There we go.
Cool.
Mm,
All right.
Yeah, Chris, you'll be interested in this one.
I know. Yeah. Did anybody make any more commentary about that one? Because oh, what is it? William had one that was like a dead match.
Well, the thing that you and I were talking about at the foray this past weekend, Igor picked it up and was like, "Yeah, I think I know what this is," and this is the name that he put to it.
Really?
Yep.
Yeah, and I checked with KOH, and it produced the reaction that I was seeking. The color change with KOH.
I took a chunk of it home to get some spores and everything, and all I got was slugs that crawled out of it eventually, which was
Oh my
super
God.
pleasant.
Needs more syllables.
Yeah, I mean, I can thank Rick Kerrigan for that memorable name. I mean, at least he will never forget it. I mean, this thing apparently just likes the pine barrens. I mean, we have Floridanus. We have Agaricus floridanus. I think another one species that transpired with sequencing, but this one is by far the most common one. Has a very pleasant kind of a licorice, chocolatey smell, and the ring is big. usually like that, maybe sometimes smaller. It's a very attractive species. I don't know if it's edible though. Probably is.
If it smells nice, it probably is.
It doesn't stain yellow with
Now, is
KOH
this in
or.
section Arvenses, do you know, or subgenus Arvenses? Or whatever?
Well, you have the book, so you can check.
Oh, yeah.
Because my book is still wrapped in its original
Hold
plastic.
on. I
Oh.
will walk to the other room and go get mine.
What do you use it as a doorstop or something?
No, I'm thinking, well, maybe if I wait a few more years, I can sell it for twice the price that
Oh.
I purchased it for because it's out of print.
What?
It
You
was an
bought it as
investment.
an investment.
Yes, as an investment, yeah. In perfect condition, unopened, unused,
I'm telling you,
and vintage as well.
I'm tired from walking across the house with this thing.
Yeah, it's gonna pull your bicep pretty well.
Why I'm jacked,
Emily,
man.
Emily, this is a really good observation, by the way.
Oh yeah, very nice. Very nice.
Yeah, this thing is beautiful.
Yeah, the ornamentation on the cap, those smoky, vinaceous brown scales are very characteristic, and they're arranged, like they're very dense in the center, and then eventually they sort of spread out. It's like an explosion kind of thing, So
So this name actually means, I guess, like
Yeah,
like
smoky.
wine-colored brown, but smoky, sort of.
Yeah.
I think.
yeah,
I was gonna ask Igor where he got those descriptors, but I know where it's in the name.
yeah, it's in the name. Yeah, vinaceous brown, smoky vinaceous brown. That's what it means. Yeah.
Yeah.
338. Man, that's a huge book.
Yeah, I haven't tried any agaricus other than the ones that I see in the store, which is by Sporus.
I used to eat what I have called abrupta bulbus, and that one was pretty good. And horse mushrooms are really good, but you just have to watch where you get them from. And most of them are not arvensis. If you look at the spores, you'll see that they're crocodilinus, which has the bigger spores.
Crocodilinus yeah, I got that one too. Actually, it was growing in my property, or it was growing. But I got that one sequenced, and that's how I found out that crocodilinus doesn't have to have a bad hair day.
No, it's sometimes very smooth, and then it gets mistaken for arvensis.
Yeah, it kind of gets dusky yellow on the top, and it also
Yep.
smells very nice, just like
So
this one.
does the same thing with arvensis. You can tell from the spore size.
Yeah, they're huge. They're huge.
Yeah, they have for agaricus, the spores are really
Right.
big, like 10 microns or
Yeah,
something.
10 to 12, something like that. Yeah.
Yeah.
It says the KOH on the Pelias surface, teal,
Greenish.
olivaceous, grayish.
Yes.
Elsewhere, sordid or obscurely yellowish, which, man, I don't know that word at all.
Yeah, but what section is it in?
Subrutilescence.
Telescence. Yeah,
Okay.
subtelescence. Yeah.
This is
So
why I
it's-
like this book, though. Listen to this description. Piptopelius, commonly dark, vinaceous brown, Brussels brown.
Russell Brown.
He's
Yeah.
so snooty.
Brussels brown.
Brussels Brown. Yeah.
They might
Yeah, I
not
mean,
even
like,
know
what a
that is
description.
at
Who
Lowe's.
talks like that?
What is Brussels Brown anyway?
Yeah, who cares?
Sounds like a name from a very old book on colors that
Yeah.
costs a
Yeah.
fortune.
Yeah, I'm glad somebody looked at that. We were going over this thing. I was just like, I was staring at it. I'm like, I feel like this is an agaricus.
Yeah, that's as far as I got. I just called it an agaricus and said I don't think I can figure it out any
You know,
further than that.
that reminds me. Could you send out that thing that you and Sue made, Igor? I
Oh,
don't know.
it's in the newsletter.
Do you know which one?
From
I
last
was
year, I
looking
think.
for it.
It was from last year, I think, maybe the last issue or the penultimate issue. But I think it's a 2024 newsletter.
All right.
I just have to flip through a few issues, including maybe the first one of this year, just in case.
Yeah, I was looking for it. I couldn't find it.
It's probably issue 54-3 or 4,
All right,
which is last
I'm
year's.
on the hunt for it. I'm gonna print it out this time.
So, and the third observation is another gilt mushroom, and that one has more questions.
Okay, hang on. There we go.
Parvin State Park. Right.
It's a little like Modesta.
How big was this?
Oh, I'd say about maybe 2.5 inches across.
2.5 inches.
So the way
Yeah.
you tell Variata is by observing forks in the gills, like halfway to the margin, near the margin. So I only see a few gills here, but I don't see any forks.
Yeah, no.
That's what I said in the comments, that I'm not seeing any forks.
No,
So
it's not
it's
Variata.
not Riata. No.
I see a couple of forks
Yeah,
by the dive, no?
Well,
No, but
a lot
it
of
should like
russulas
fork.
have forks in the gills near the stem, but Variata has forks that are close to the margin, halfway to the margin. Sometimes forks again.
Basically, yeah, there's a lot of fork in Riata, and it's like forks and forks. The gills split, and then another gill that was just splits again.
Yep.
Gonna-
But this one has forks near the stem, but a lot of
Yes.
them have that.
Right. It's a common feature for a lot of russulas. It's not
Mhm,
an indicator of anything.
This looks like Modesta to me.
Yeah, except that it's kind of a robust.
Yeah, they can be pretty big.
Yeah, I usually see them when they're small,
Yeah.
maybe like 1.5 inches at the most.
I see them much bigger than that.
Yeah, I think we found Modesta in the Pine Barrens before a long time ago. So that
Oh.
species is there. Yeah, and they do sort of become kind of a yellowish brown in the center.
In the center, yep, and
Yeah,
gray towards the margin.
when Modesta is very young, it's covered with this sort of a
Bloom.
dusting,
Yeah.
pulverulence, which is whitish, like a bloom. It's like a whitish bloom.
Yeah. Yeah, it looks like it was dusted.
And that's one of the telltale characteristics of Russula modesta. And at that time, it's completely grayish green. And I think when they get bigger, when they mature, you start to see those colors that are sort of more maybe similar to Cyanoxantha, which is a totally different species.
Yeah, Cyanoxantha actually has like bluish tints.
Yes, blue tinge is correct. Yeah.
Yeah.
So this
Well,
one is
that's where
green.
the name comes from, I guess.
Yeah, correct.
Cyanoxantha.
Which means yellow, and blue. Yeah.
Yeah.
Interesting.
Yeah. Modesta, I think, is a good proposal.
Okay.
Yeah, that's what I would guess. But russulas are tricky. You can get fooled by them a lot.
I'm pretty sure it's mild tasting and it's going to peel maybe halfway through.
Yeah, I don't know how much it would peel, but it would be mild tasting.
Yeah, it was not spicy. I did actually taste it.
Yeah,
Right.
they're good edibles. They've eats them.
Yeah, they're pretty. When I find young ones that are firm and no insect damage, they're pretty good. There's a few other russulas I eat that are the mild tasting ones that are firm. A lot of the russulas, they're just there's not much to them. They fall. You pick them, put them in a basket or a bag. By the time you get home, they fall apart. But this one's a little bit more robust and resilient. Well, Modesta is at least, and I think that's probably what this is.
Okay, that's another thing that hasn't been found much or put on INAT that much from this area.
That's because people are reluctant to propose species names for russulas on
Right.
INAT.
Right. Well, that's why these groups are helpful.
Right.
Yeah,
So,
and that's one thing I like better about Mushroom Observer. You can put this on Mushroom Observer and say like, oh, this could be Russula modesta.
No, I do
you're
wish
not
you
going to get a whole lot of argument on that. But on INAT, it's you put Russula modesta, and you're basically saying that's what this is.
Right.
And then no one is going to hop on there to agree or disagree.
And then it just sits there like that. Yeah, that's there's drawbacks to both systems, but that's the shortcoming on INAT.
So Chris unearthed that article that Sue put together on Agaricus. It came from issue 54-2, which is the second issue of the year, second quarter. So I was wrong about it being in the third or fourth. And there's a nice table there on page 13 and 14. So if you want to brush up on your basic Agaricus identifications, that's a good article to go to. Has some names there as well, descriptions, sections, staining and chemical reactions, and so forth.
Yeah, that's an excellent resource. I'll be using it.
So apparently the only member of section Subrutilescens that we have on that table is Vinosa humidus. So, and quite a few from section Arvensis. Abruptibulbus, Crocodilinus, Nanagustus, Silvicola, and Sebrifescens. Yeah, all right.
Cool.
Igor, did you ever solve the issue of being able to propose another name on Mushroom Observer, or is it just being tricky tonight?
Basically, I emailed, I reported this as a bug, and I got a response from one of the administrators, Joseph Cohen, who got back to me pretty quickly. He said that he didn't have time to look at this, but he forwarded that ticket to to the programmer. They have a good programmer now. But he said there's a workaround. You have to right-click on that button. So I use Chromebook, so the right-click doesn't work for me. It's like a double tap. You have to tap in succession twice, but not with the same finger. I do it with 2 fingers, and then it opens up a sub-menu, and you select open another tab. And when you open another tab, you will basically be proposing that name in a separate window, separate
Oh, it
tab.
worked.
Yeah,
It worked. Thank you.
So it's a workaround. It's a temporary
Yeah.
workaround, but it works.
Oh, you're talking about proposing on Mushroom Observer?
Mm-hmm.
Yeah.
Yeah, because I clicked before on propose a name and nothing happened.
And what browser are you using?
Chrome.
Chrome. Okay.
Well, so am I, but it did work.
What's that?
I'm using Chrome too, but it did work.
Oh.
The open
Well,
and
maybe it'll work on your own observation.
No,
Oh, well,
the workaround works for her, right?
Oh, I'm
that-
sorry. I didn't hear about the workaround. I took a
yeah,
look.
Yeah.
the workaround should work. So I thought that maybe this is going to be something browser specific, but sounds like it's not.
Yeah, just right click and open a new tab right over where you were going to click if it's not working. And
Right.
that's
Whoa.
So
Right clicking makes me nervous.
this workaround was there was another problem some time ago, and I think it was a similar workaround. You could right click on something and open a different tab or a window, and then do your thing there.
Well, but whatever it is, they'll fix it.
They'll fix it, yeah. But I'm just wondering if they actually get bug reports from other people, because I've sent them quite a few, and they're probably tired of me.
No, they sometimes they'll notice things that I didn't notice, and they'll ask me, and so they want you to send as many bugs as you can find.
Oh, I will. I mean, I think they encourage me, but I was just joking that like
yeah,
not
okay,
like not again, not this guy again with another bug.
yeah. Joe Cohen is really good. He's really he's very dedicated.
Oh yes, he is. Yeah, we have a good relationship there.
He's a really good guy.
Yeah,
he does a good job.
Yeah, they have a programmer named Nemo from I guess Australia. I never met him or saw him, but he's quite good. So he's been doing a lot of the changes that are behind that you see on the mound behind the scenes. Finally, they were able to hire somebody and pay that person a salary. Because now they're getting donations, and they're certainly going to get a donation from me this year again.
Oh yeah, I should send them a donation.
Yeah, I send them money every year. So
yeah, our club sends them money too, because I use it for a lot of our club functions. Yeah, that's coming up now. We'll have to send them some money.
Yeah, I usually send the money around Thanksgiving.
Mm-hmm. Good. As a club, I think we probably have money for a donation there as well.
Yeah, absolutely. I think we should do that. I mean, it doesn't have to be like a gargantuan amount, obviously, but I think any good amount helps.
Yeah, agreed.
I'll talk to Lyle about this.
Do we have anyone else that is sharing mushrooms?
That's what I was just gonna ask.
Well, I did send those emails in. I mean, if anybody wants to look at them, we can. It's getting a little late, but I'm still not sleeping, so I guess I guess I can talk about mushrooms.
And then Byron's going to come in, bite your hand, and howl.
After Karen goes to bed, he behaves
Okay.
while she's here.
You sent some more to Taxonomy Tuesday?
Yeah, I sent. Well, there was an email I sent. You actually brought it up before, but then we moved on.
Yeah, we haven't looked at.
Yeah, we brought it up, but I deferred to Hunter because she was in a hurry to get ready to travel or something.
Yeah, there we are. That's right.
So, a common polypore, but I thought I'd share this because of the name that is proposed is a new name. So I think Igor actually proposed this name, and I'm going to send it into Mycoblitz. It was found and collected within the time parameters, so I can get it sequenced for $3. Well, hopefully sequenced. We'll see. But I think this is worth sequencing because this Hispididoides or something, Hispidus or Hispidoides. It's a relatively new name, and I looked on GenBank to see what was available in terms of sequences, and most of them come from the Western United States. So I'm kind of wondering if there still might be some question as to what the Eastern species is. Is it the same thing?
I think you have to go to the paper and see what they say about the distribution of Hispidoides.
Yeah, that would. Yeah, I don't have the paper. Oh, wait. Did you post it?
I think it's posted. I think I was able to get it somehow.
Oh.
But
it's not. You didn't put it in the observation, though.
No, but if you look at maybe the name, if you click on the name itself, maybe there's a link to the.
Okay. Yeah.
You have to go below. No. You have to
I'll look around.
Scroll down and then click on the name. that's not even sure. Like,
Yeah.
where's the
Well,
rest of the observation?
Well, that's all I got.
oh,
There should
that's
be like a
just the
name
photo.
proposed.
There's a photo of the underside also, and
No,
I
no, no.
didn't.
But you should be able to see the proposed name
Oh, the proposed. I'm sorry. Right.
the below. Yeah. So that's
Yeah.
I'm
Chris,
I'm
are
not, I don't
you?
I
understand why
don't.
you can
I don't
explore
have
that more.
mushroom. I'm not on Mushroom Observer.
Yeah, you got to log in, right?
Oh, okay. Never mind. Okay, never mind. Okay, got it.
Yeah. When you rub the bottom, it bruises brown, greenish brown. It's already kind of green anyway, but it does bruise darker. So this is what we used to call Phellinus schweinitzii, but I guess
Yeah.
that's European.
Oh, so is that the idea of why you're not calling this Schweinitzii? It's proven that that's solely European.
I think
It doesn't
it's a
exist here.
relatively new hypothesis, and I guess there's some data available to back up.
Yeah. Well, basically, as far as I remember from that paper, all of their accessions of Schweinitzer are from Europe.
Right, but it's that doesn't mean it can't be Schweinitzii, or it might be something else. So I'm gonna, hopefully I'll get a sequence for this.
Yeah, that's interesting.
Yeah.
Because I would have just seen it and said, "Yeah, that's Schweinitzii."
Well, that's what I did. I
Yeah.
just put it up as Schweinitzii, and then Igor proposed, and then I remember, because I actually knew somebody had told me, maybe Igor told me, I don't know, somebody had told me there were some new names for the Dyer's polypore. So anyway, it's a pretty good specimen. It's a nice, it's young, but it's not real young. The pores are open. I didn't get any spores. I actually hung it in a window. I was just going to dry it out to give to like Susan or Cheryl Dawson or somebody, and
Right.
and then
So
after I saw this discussion, I hacked off a piece and put it in the dryer. So hopefully it'll yield a sequence.
yeah,
So I've said
it,
things to
it's
Steve
still
Russell that really
it's,
you would think should not yield a sequence, and sometimes they do. I mean, things that just sat on a table for 2 days, stuff like that, and sometimes he still gets data from it.
Well, the paper is actually downloadable from ResearchGate. I posted a link in the chat. So you can basically download that paper as a PDF and read it.
Sounds good. Yeah. I just copied it to my clipboard. So
yeah, it's from the Journal of Systematics and Evolution. It's apparently one of those journals that are you have to, as an author, you have to pay to publish, so it's freely accessible to the public.
I see.
Yeah.
Okay.
As
Well, I
opposed to
just
something like Mycologia, where authors don't pay anything, but then you have to subscribe or purchase an article for like $65
right.
for like 2 days. Yeah.
Huh.
They also dry out real well in a cardboard box in a real hot car.
Well, not a highly recommended technique, but I guess it works.
So I'm looking at the tree in the paper, and the accessions or the samples, vouchers came from Michigan, Pennsylvania, Wisconsin, Connecticut, New York, and Alaska.
And those are all the hispidoides.
Hispidoides, correct.
So that's probably what I have then.
And there's one from Montana here. So yeah.
Okay, because I didn't investigate a whole lot. I looked at like 3 sequences on GenBank, and and they were all from like California and Washington or whatever. So
Probably,
it made me
yeah.
wonder,
Well, could be just like recent accessions from INAT or whatnot. And interestingly, the closest relative is the species that they collect only in Arizona or the Southwest. It's called Occidente americanus, and that's the closest relative to Hispidoides.
Okay. Well, here's something that I found for the first time. I have not found this before. And it's been very dry here, after a very wet July. August has been dry. So when it gets really dry and I want to look for mushrooms, I go to sphagnum swamp at relatively high elevation, probably about 2,300 feet above sea level. And there's a very large sphagnum swamp near something called Lake Jean. And I find interesting things there. So Microglossum fumosus, I think is the species name. And so it looks like fumosum. So it looks like what it's supposed to, and the spores are the right size, and the paraphyses fit the description of what they're supposed to look like. So I'm pretty sure it's what I have here. But I'll send some in. I'll send some into MycoBlitz.
Dave, first of all, what was the date on the MicroBlitz? I wasn't even.
It was I think the 9th through the 17th
of August.
Yeah, it just ended on Sunday. Sunday was the last day.
Yeah, let's look at these spores. Cool.
Yeah,
this is a cool
they had
find.
like 9 to 11 septa, and that's within the range of what is reported for the species. But the lengths were pretty much what you would expect. I think like 25 to 37. I think I may have put, but I think that's what I estimated.
Oh, look at
So,
that!
and there's an ascus.
Mm-hmm. Cool.
And I got some photos of paraphyses also there, and that's a pretty good photo of an ascus there. And that one's not bad either. You can see what's going on inside it, sort of. The spores are really jammed together though, so it's hard to tell what the individual spores are. But there's a paraphys, so it's like bent at the apex, and it's a little bit like tapered, inflated, sort of. It's like it gets thicker progressively towards the apex. And there was one I found that was sort of capitate. And if you look on MycoQuebec, it's what you see. There's photos of micro on MycoQuebec, and it's pretty much what you see for the paraphyses. There's another paraphys, and there's another one. I have one more, I think. It's that one's sort of like capitate. So I'm pretty sure that's what these are, but it was something I never found before. So I was kind of excited to see it. I guess it's not really all that uncommon, but it's just something I have not found until.
I've never seen it either, Dave. I'd be excited to find it. When you say it's not uncommon, what you have
I don't
seen?
know. It's I think it's on both MycoQuebec and Mushroom Expert. I'm not sure. So, so I'm assuming then that but it might not be common. I forget. I forget what MycoQuebec said, because they always say like how common they think something is. So I got a nice collection of these. I'll send one to Steve, and I'll save the rest. And we're hoping to be able to get stuff stored in the Philadelphia Museum of Natural History Fungarium. Okay, so this is a good one here. Entoloma species PA02 I was one of the first people to find this, I believe, in this same location, and that we found it in a different location as well. It's an Entoloma. Look at this little thing as an Entoloma. It's been sequenced a few times, and I'm not going to have this one sequenced because I pretty much I know what this is. I've had this sequenced a few times, and a few that were picked in the exact same habitat location have been sequenced. They look exactly like these two, and so
Look at that.
Pretty cool little mushroom. When I first found it, I didn't know what it was.
Yeah.
I-
White thing with a pink spore print. It's an Entoloma, right?
Well, I didn't even get a whole lot of spores the first time I brought it home, but I did look under the scope, and once I looked under the scope, I knew what it was because the spores are most definitely Entoloma spores. So I wanted to get it in the dryer as soon as I could because I wanted to preserve it. So I just hacked off a very tiny piece of gill and smash mounted, and I got some spores. So when you get spores from a smash mount, it's kind of there's stuff in the way. You don't get a real good seal where everything's in one virtual plane. So the details kind of get lost a little bit. You can see those are Entoloma spores. So that's
a cool find. What was the name?
Pennsylvania species 02. It has no name yet. I mean,
okay.
I didn't send material to Tim Baroni, and I asked Steve Russell if he had any left, that if he could send it to Tim Baroni, but I never heard anything after that. But judging from my brief discussions with Tim Baroni, who's the North American expert on Entoloma, he didn't know what it was. So
yeah, I found something similar in sphagnum recently, and Jason sent me some papers because shoot, are we still recording? We might get a little personal on this. I don't want to share too much information.
Yeah, it is still recording.
Yeah, a friend of a friend sent me some stuff. I'll send to you, Dave.
Oh, okay, thanks. So, what the sorts of things I find when there's a drought and I go up into the sphagnum swamp, I find stuff like this. So this is probably Galerina pelludosa, would be my guess. The spore dimensions fit. The velar remnants on the stalks are pelludosa trait. The habitat, sphagnum, that's where pelludosa grows. I've had pelludosa from the same location and habitat sequenced by Steve in the past, and it was what it was. But you can look at, there's almost like a little ring on the stalk there. It's a, I don't know if they call this partial veil or universal veil. I'm not even sure, but they call this stuff velar remnants. So the next photo, I think I may have tried to take a photo through my, or the next one after this, through my, no, I guess I didn't. I tried to get a photo through the binocular scope, but it didn't come out. These were mounted in melters, but this scope that I use here to estimate measurements doesn't show color very well. But they darkened somewhat in melters. So I'm wondering, what constitutes, inactive in melters as opposed to weakly dextrinoid? Should there be some sort of distinction there? Because these pelludosa spores, and I think these are pelludosa, and in the past I've looked at pelludosa spores mounted in melters, and it seems like they darken a little bit, and some of them get to be a little bit reddish brown. And so I call it weakly to moderately dextrinoid. But if you look at Smith and Singer, they did a big monograph on Galerina. They say that pelludosa, what they say is the spores don't look that much different than what they look like in KOH, but they do say that they darken somewhat. So that actually kind of fits what I've seen when I mount them in melters. What I need to do is get some more of these and mount them twice. Put some in melters and put some in KOH and then compare what the colors look like. Just something else to try to find out.
Yeah.
Probably poisonous. It's a Galerina.
Cool observation though. Appreciate you
Oh, sure. Thanks.
looking at that.
So, generally speaking, my concept of Calabiopsis confluens is a mushroom that usually has a very pale cap, something almost white. But these really otherwise look like Calabiopsis confluens, and they were growing in sort of small clusters in litter, and they're pretty big. So there are other Calabiopsis species, and these are things that used to be gymnopus. But there are other Calabiopsis species that have hairy stalks that grow on litter or on wood, but they're usually smaller. And Decora grows directly on wood, usually fairly good sized pieces of wood. Biformis and subnuda are smaller and not quite this hairy. Calabiopsis confluens has a very hairy stalk, at least for a while. Maybe that erodes, but you can see here, these stalks are very hairy. They were all like this. So I think Calabiopsis confluens. I'll send it in. Another feature, and I think there's one more photo here that this is through my binocular scope, so the resolution's not real good. But the thing that's interesting about this photo is the spores look blue. These were mounted in cotton blue, and the spore walls absorbed the blue stain. So a lot of times you'll see on Mycoquebec under description of spores, they will use the word cyanophilic, CYANPHIL. Wait, PH cyano. Oh yeah, IL. I see. Cyanophilic. I looked up the word. It is a word. it's used in the same way in biology for like tissues that absorb blue stain. So, is that a trait of this species in particular? I don't know. I think I looked it up, but didn't see anything about the spores being cyanophilic. But they were pretty, they turned pretty blue, mounted in cotton blue.
So what do you
So
just
anyway,
you stain them and wash them out?
a little bit, yeah. I'll put well. First of all, I dilute the cotton blue pretty much. Straight up cotton blue is like so dense and thick and when I first used cotton blue, I just used it straight. It's like you couldn't even see anything. It was so dark. So yeah, I put a little cotton blue on there, and then I'll put a little distilled water on and blot some of it off. And and then the tissue, of course, is what you want to see if it absorbs the stain. So I think I'm doing that the right way. I'm not sure. A lot of what I do, I've sort of just invented on my own. Now this is the same mount here, the spores through the other scope. The other scope doesn't pick up color very well. It's an old scope. It's an old monocular scope, but it's useful because I can see shape and I can get estimates on dimensions. Very tightly spaced gills. That is a trait of confluens. And when confluens is very young, if that's what this is, Calabiopsis confluens, the gills are so tight it looks like a smooth surface, kind of like Lactophlus piperatus, same kind of thing. But these are pretty well expanded, and the gills are still very close. So I think so, but it's an interesting one. You know, I'm going to send it in for sequencing. We'll see if we get any result. But the closest thing I can think of is Calybiopsis confluens.
Nice. Oh,
I guess that was it. Was that I guess that was 5.
I think that's it.
All right. Thank you.
Thank you.
Sure. You're welcome.
By the way, seems like the create name button on the Mo is working. Nathan Wilson pushed out a fix for the problem, so I just checked and it's working for me too. So that came about very quickly, thanks to him. He said that he didn't test it extensively, but for him it worked. So, so it looks like it's a quality solution to the problem.